Okay, we are ready to start. People, don't be quiet. I'm going to have you hold your hand out and wham. You know, they did that to me in 3rd grade. I got in a fight out in the play yard. The rulers, they used to do that. Do they do that anymore? Probably not. They did it. I still remember the sting. Who brought Maverick yesterday? I forgot to give him the prize.
Oh.
Pass that back. Pass the penis back. Who laughed at that? And then Taylor, I'll have to give Royal his treat because I order those. I spend money on those bull pizzles. They're officially called maybe. Because if somebody thinks enough of us to bring in the animal, a dog especially, then I have to, reward them with some kind of treat, right? It's only fair. Let's see. So I want to talk about dogs a little bit because we have a dog coming tomorrow, and part of my deal is I always want to train people in how to interact with dogs. Some of you might have way more experience than me with dogs. I kind of doubt it. I'm older than you. But some may have no experience. So here's where you learn. Yesterday, those 2 dogs were probably the best examples of dogs that wanted to interact with people, right? I mean, Maverick was going voluntarily over in the aisles, halls, whatever you want to call it. Royal was more wanted to be by mom, but Royal enjoyed interacting with people, but he stayed here. And then you noticed how when we unexpectedly had them both here, that we kept them on a leash and let them have visual contact first, nothing tactile. No growling. They saw each other. That was a good sign because they both kind of said, oh, this is boring. This is another black dog. So then we got them closer in a controlled manner. Nothing was happening, nothing was happening. So we're at 0, means we're like neutral. They went by each other, they smelled each other, no big deal, so then we let them loose. If they would have came in and had a disagreement right away, then we would be below 0. We would be in the negative category, and we'd have to get them back up to 0 to start interacting with them, right? So never let 2 dogs like that have a misunderstanding at first. If one would have growled at another as I was coming down, then we would have kept them probably both on the leash. That would have been fine, whatever. Okay, so that was great. Everybody interacted with it. Tomorrow we have a timid dog coming, I understand. Maybe timid sometimes it's unknown how a dog reacts to a big room with 100 people in it, right? So here's what you do with a timid dog or a dog you don't know. Totally ignore it. Ignore it. You don't interact with it. You come in, you sit down, and then you see what the dog is doing. Its body language will tell you. If it's off in the corner with mom or sitting by mom, don't go up to it and pet it. You totally ignore it. Let it come to you, and then we'll just see how it happens, right? That's how you do it. That's the best advice if you don't know the dog at all. Don't walk up to it. Last fall, we had a dog, a Mastiff, in the center there, and I walked up to it, and it growled at me. And was I afraid? No, I wasn't afraid because Nala just said, Rod, I don't want you walking up to me like that. It was like a nice little growl, if growls can be nice, whatever. So I knew that. Well, then what I did during class is I backed up to it, and I was talking like this, and it didn't growl at me at all. It didn't want me to come up face to face to it, okay? So, and I wasn't like, oh, we gotta get that dog out of here, everybody's in danger. No, it was a dog who was walking around too. It just didn't want me to walk up to it. Might have been a male thing, right? Some dogs are biased against males or females, whatever. Who knows? So tomorrow, ignore the dog, and then you'll see what I do with the dog, and then you can play it after that. Okay, anybody have any questions? Here we go.
Okay. So
Jams.
Hello. You said that.
No, wait a minute. Jams and I were comparing tans yesterday.
We
right?
were.
And she's got
He
a
won.
better. No, you have a better tan than I do, but I'm working on it. Okay, go ahead.
I'm just kidding. No. What was I gonna say? I totally forgot now. Look what you did. Oh, so you said that like if we start like with a misunderstanding, And
With the dogs now you're talking
yes, with the dogs, sorry.
Not you and me.
No. And you start like at below 0 in the negative,
Yeah.
so you have to get it up to 0. How necessarily do you do that?
You may not be able to do it.
Okay.
You can just let time go on, and then let's say they would have growled at each other, then we would have kept them on leash and probably brought them a little closer together over time.
Okay.
And we may not have enough time to get to back up to 0. You know, the dogs weren't living together. They weren't, you know, they may never see each other again. So if we were below 0, that means we just keep them apart. But we would have maybe got them a little closer if we would have had time.
Okay.
Yeah. And who knows how that works. Of course, with animal behavior, nothing's 100%, right? You can always say, never run from a dog. And that's usually good advice, 99.9%. Never run, because they have this innate thing, when something moves fast away from them, they want to chase it, don't they? They do. And if you get a pack of dogs, for sure never run, because they're going to give chase. Too many stories about little kids getting mauled or killed by 2 or more dogs, right? A lot of stories like that. Okay, other questions? Anything? We didn't get any email questions, that's fine. I've got a few things on my list I want to do. And maybe what I'll start with is I want to make sure everybody understands diffusion, osmosis, edema, and I'll introduce lymphedema. I think that might be in the videos too. And a little thing about how tissue flows in fluid, because it's very important. I think the video did talk about, the videos I should say, isotonic, hypotonic, and hypertonic. No or yes? Some are saying yes and some are saying no. I'll review that a little bit too, because it's all about osmosis and then physiological saline, right? So let's start with the document. Well, let's go to my textbook. And let's see, we've got Google, and I'm going to do osmosis. SIS. Okay, here we go. Now remember, this is your textbook. You can do images, and let me start with diffusion, and then I'll go to osmosis. Diffusion is always passive. There is no pump. There's nothing active about it. So it's going to obey the laws of physics. Now here's the deal. I remember somebody did this to me when I was in your shoe someplace. Somebody said, okay, I'm going to drop some perfume in the back corner. And then in a little bit, we're going to smell it up here. We don't smell it right away. But that's an example of diffusion. That's a lie. That's a total lie. If there was some perfume back in that corner and you relied on diffusion, you would stand up here for 1,000 years and may never smell it. Okay? So that's not diffusion. Diffusion only occurs over very microscopic distances. Okay? Now, you will agree that we will smell the perfume up here, right? Probably. What mechanism is transporting the perfume if I smell it up here after it's dropped there for a while? And I'll give you a little hint, a visual hint. Okay. Good word, yeah, okay. Airflow. And whenever air is flowing, and you can see there's vents in here, right? If we had a little tissue up there, I could show you there's air coming out of there. And just us moving is making air flow, right? And that's called convection. C-O-N-V-E-C-T-I-O-N, convection. Convection conveys the perfume up here, not diffusion. Diffusion is microscopic. So we rely on diffusion for oxygen to get from the alveolus lumen, right? You know what a lumen is, L-U-M-E-N. That's any open area in a tubular structure. So air, oxygen, is in the alveolar lumen, and it diffuses across the diffusion barrier into a red blood cell. And that's microscopic. I don't know how many microns it is, but it's not many. You take a breath, it does that. Carbon dioxide flows the opposite direction, right? We exhale carbon dioxide. That's diffusion. Now, osmosis is the diffusion of water. So, osmosis is water moving. And there's always these great examples, and this one's not bad right here, that you have a semi-permeable membrane. Semi means some things go through, some things not. In this case, none of the little blue or purple things can get across the membrane. And think of these as red blood cells. Think of this line as the wall of a capillary. And capillaries are leaky. You can write that down. Capillaries are leaky. That means it wouldn't make a good garden hose. Things can flow out and in a capillary, and especially water. Red blood cells cannot leave the capillary. The holes aren't big enough, and platelets don't leave unless, of course, there's some injury or something. And then in previous discussions, I talked about how white blood cells can squeeze through these little pores. They can leave a capillary or go back in. It's amazing. We talked about pus yesterday. What is pus? Pus is white blood cells leaving the blood vessel, going through a capillary wall and getting into where the bad guys are. They're actually called to that area. So in this simple example, there's a semi-permeable membrane. Let's see, did they do that right? There's 7 dots over here, there's 7 over there. That proves they're not moving. But water goes from high concentration to low concentration. That's what diffusion is, right? Something is high and it goes to low. So on this side, there's more water concentration. And so water diffuses to this side. Initially the levels were equal, but now the level goes up here because water is diffusing from the initial high to a lower area. Water would never go this way. Okay, so then we get a difference in height, and if you would have put a gauge on that and would have like measured the force of water coming up, you would call that osmotic pressure. Like if you would have had a little something on there that could measure some tiny little force, you know, the water went up, right? So it would have some force on it, and you could actually prevent the water from coming up, and you can measure osmotic pressure. So very important concept. The number of dots never change on either side because they can't move through that membrane. But this is a great example of a capillary. They're leaky. Because capillaries are like this. Here's one cell of a capillary wall, and here's another. You can never make it waterproof, right? And then another one comes here because capillaries are built that way. You put a cell here, you put a cell here, you put another cell here, you put another cell here, now you've got a tube. Because when an animal grows, they must make capillaries, right? A new tissue. Even tumors do this. Tumors are wicked. They send out chemicals that says, hey, start building a capillary here, right? Every tumor has a blood supply. If it didn't have a blood supply, it wouldn't be living. So it's an amazing process. Capillaries are leaky, but there's one exception. In the brain, capillaries tend to be tight. They're not leaky in the brain. And Mother Nature did a great job because then if capillaries are tight, less chemicals can get through, and it protects your brain from chemicals. There are exceptions like anesthetics, right? If you go do surgery, you either inhalation, anesthesia, the chemical gets in your blood, it goes all over, and if you're under anesthesia, that chemical had to get across the brain, into the brain. So in the brain, since we're talking about capillaries and all this stuff, it's tight, and it's so tight, somebody labeled it the BBB. That's all capital letters. Anybody know of the BBB? The blood-brain barrier. In high school biology or someplace we had a beginning freshman biology here, whatever. Blood-brain barrier. It's tight. Very few things can get across. And it's not like cellophane. It's not like you wrap the outside of the brain with cellophane. No. The blood-brain barrier is every interface where there's a capillary facing brain tissue, right? So brain tissue, capillary, blood in here. That's the blood-brain barrier, that capillary wall from the brain. So could you ever put the blood-brain barrier on the table? No, because it's at every interface between the capillary and the brain. Could you put a lung on the table? Yes. It's not distinct like a lung or a kidney or anything like that. I could bring a horse aorta. I should bring a horse aorta sometime. You should see it. It's bigger than my thumb. And it's really thick wall, because through the arteries, these large vessels, nothing is getting exchanged. All the work is done at the capillary. Okay, so then that's osmosis, capillary. That's good. And then now I'm going to do edema. Because it's all kind of related and we're throwing things in like blood-brain barrier. But it's okay. Here's one great example. I mean, when I say great, you know, I just mean it's for us to learn. I'm not happy that the person has that. That's called pitting edema. You push your finger in it, and then you remove it, and your impression of your finger is still there. Pitting edema, that's 2 words. But you can obviously tell that's not normal. Because in most tissue, you press your finger in there, and it springs back right away, right? That shows you pretty nice. This shows you edema. And yesterday we said edema is excess interstitial fluid. There's fluid around every cell it's outside of the blood vessel. But every tissue has fluid in it that's not in the blood, and that's called, and the fluid that's between cells is called interstitial fluid. Or you might want to call it intercellular fluid, I-N-T-E-R, intercellular fluid. And of course, every cell has fluid in it, and that's intra, I-N-T-R-A, intracellular fluid, right? So there's intracellular fluid, there's extracellular fluid, that's another thing you could call it, extracellular fluid. Then there's blood, right? And if you squeeze all the fluid, all the water out of a horse, you have 65% of its weight as water weight. That goes for us too. There are exceptions to that, like embryos are 95% water. And old people are less water. So we always say aging is a dehydration process, right? You start out at 95% water, and when you're really old, I'm not sure what the percentage is, 60 or whatever. Anyway, you lose water. Okay, so edema is excess interstitial fluid. Oops, I lost my thing. Way back here.
Did you say the embryo was 90 or 95?
Oh, 95. Don't ever quote me on that. I'm not sure, really. It's close to that.
Okay.
Something like that. That's what textbooks often say. Okay, let me get back here. Okay, so now let me do lymphedema. That's all one word, lymphedema. Yeah, and I should draw a picture. Let me do that too. Let me do that. And I know it was in one of the videos, but just so we make sure we know it. So how does this fluid get excess? And what it is it's not excess fluid getting into the tissue, it's not enough fluid being drained away. So let me draw here on this thing. Okay, so is this showing up? Yeah. Okay, because I'm looking here, I'm not looking back there. So here's a capillary. I'm trying to make it look like there's little pores between them, little holes. Right, blood is going to flow this way. Just to make sure there's red blood cells here. Right, you guys are seeing that? Yeah. Okay. So then, this is the in the lumen of the capillary. Right, I've got a cropped little section here. I can't draw 3-dimensional, right? So there's inside the capillary. This is outside the capillary. Outside the capillary, there would be cells. I'll put a nucleus in there. And if blood is flowing this way, if it's coming into the tissue here, then this is an arteriole. That's on the videos, I know. You have an arteriole, is the last blood vessel before you get to a capillary. Then you have a venule, right? So then, I know the video shows fluid's going to move this way. So I'm going to say 100 drops get put out of the blood vessel into the tissue over time. I don't know if that's an hour or whatever. And here's the kicker though, only 99 go back in. Now that's okay if you have a checking account. If somebody puts $100 in and you spend 99, then you've got a balance of 1, right? And then a little later, if somebody puts another 100 in and you take 99 out, you got a balance of 2. That's good for us to get a balance above 0, but that's not good if that stays in here, right? Over time, you'd have one drop, then another drop, right? And that's what edema is. Over time, if this was left to be, and this is normal, if that was the only thing going on, everything would become edemic. So we have to take care of the extra drop, or the extra mil, whatever you want to call it. So we need another pipe. So, and these pipes start out with a capped end. This is a pipe that starts out with a cap on the end. Now how do I do that? Well, okay, I'll make a little thing like this, and then this, and I gotta put a little one-way valve in here. Fluid starts going this way in this vessel. Maybe the dots mean fluid. And every once in a while there's a one-way valve. What is that vessel called? A lymph vessel. And see, remember the Chinese proverb, hear and forget, see and remember, do and understand. So you should be able to explain this to somebody from scratch. Get a scratch piece of paper, have a study buddy and say, I'm going to show you how interstitial fluid is formed, how there might be an excess, and I'm going to show you how the excess is removed. So that one drop sooner or later gets into the lymph vessel. And notice it's capped on the end. It's not a tube. Lymph does not circulate in the body. Blood circulates, right? Comes in, goes out, whatever, blah, blah, blah. Lymph just starts here and goes that way. It's like a garden hose with a cap on the end, but there's little holes for excess fluid to get in. It's like a drain pipe, right? That's what lymph is. And if this lymph isn't working right, this lymph vessel, then you have lymphedema that I just showed you. Because that's a name of the lymph isn't working right. The lymph vessel. Okay, so there's other reasons for edema though. This pipe might be working right, but there might be too much fluid flowing out. Okay. Anybody have any questions on that? So that's lymph, osmosis, diffusion, all that stuff. Okay. Anybody have any questions? Because I wanted to show you on one of the how to get notes off the quizzes, and I'm not sure if people are doing this. The online quizzes are definitely modeled after tomorrow's assessment, right? Those, there's 6 choices, and once you get a right answer, you should take that right answer and put it in your notes. Now, if you're doing the atone thing, you could put it on whatever page it would be appropriate for, but I want to show you how to do that. Now, the thing is, none of these quizzes were made especially for you this week. They were made over time. Every semester is different. Notice how we had a little discussion on the fibrin clot last week. I don't think I mentioned that in this class for years, but somebody had a question on it, and it might be on a next assignment, right, or a next assessment. So every quiz has some answers, some questions out, like, where did this come from? You have to say, probably, it doesn't apply for us this semester. So you know what we do in this class, you know what the videos have, and you know what's the reading. That's the material that's on the assessment. Sometimes a quiz might have a question on something that we never talked about. Every semester is a little different. And sometimes the quizzes were made over time. I was telling somebody this yesterday after class. So sometimes the version doesn't tell you that maybe it lets you review it or something, because when I first found that program, I had a trial copy. And sometimes a trial copy doesn't have all the bells and whistles that if you buy it and activate it does. It's a long story. But anyway, over time, they're accumulating. I've had people help me with these. It's amazing. Let's just do this one right here. Because I want to, and see, some of these are some early ones where it says there's not a bucket. Oh, sorry, thank you. I'm looking at something and you guys aren't seeing it yet. So let me show you where I got that from. Okay, so, warm up, guys. Here we go. So I'm going to do blood, heart, lung, one. Yeah, question?
So when you're talking about interstitial fluid accumulating outside the capillaries, is it important for there to be at least some
have to, yeah, there's some there, always.
Right.
All those cells are bathed, it's the excess you don't want.
So it's because like 100 goes in and then like 99 comes out.
Yeah, no, see, did you notice how I put the 100 going out by the arterial?
Yeah, sorry, that's what I meant.
Yeah,
Yeah.
and then see, the thing is, that's high pressure, arteries are high pressure. And then it goes back near the vein wall because veins are low pressure, so it allows the fluid to come back. Yeah, it's gotta be that way. All the cells have to be bathed in fluid, there's a certain amount, but you don't want the excess. Question back here.
So this is a little bit unrelated.
Sure.
But so when you talk about infants that are born early, they have like surfactant cells.
Mhm.
Do they lack type 2 cells or are they non-functional?
Okay, yeah, and that's fine, it's good question. She's asking, I'll repeat the question because people may not have heard it. We're talking about the surfactant, the fluid that keeps alveoli inflated or reduces the surface tension of water so they can kind of inflate once a breath is taken. Any premature animal, babies in the hospital or animals, the last organ to ever develop fully is the lungs. Because the lungs are full of fluid during the fetal life. They're not used at all. You can take the lungs out of a fetus and there'd be really no consequence. Because the placenta is acting as the fetal lungs. Oxygen is diffusing across her mom's blood into the baby's blood across the placenta. So the lungs are the last. So Mother Nature said, well, I'll just fix that thing last. So very near the end where the birth is coming up, maybe 1 or 2 weeks before, then surfactant starts getting synthesized. And it's not going to inflate the lungs until the first breath, until the baby comes out or the calf comes out and takes a first breath. Then the lungs get inflated and hopefully they stay inflated. Now a premature infant comes out and the lungs aren't full of surfactant, so they take a breath, but then the alveoli go back again, back. So then I think in the video it says laboratories can synthesize it. I think the last time I looked, I can't remember, was it $500 a mil or $1,000 a mil or whatever it was. And you put that down the trachea into the airways. You actually wouldn't inject it any place. It's got to be in the lumen of the alveoli. That's what happens to premature babies. And if they know something's coming fast, there's an injection you can give the mom that tells the lungs to start making it, which is neat. Okay, does that answer your question, basically? What would say it again?
My question was, do they lack type 2 cells or are they non-functional?
No, they're just not functional yet.
Okay.
Yeah, they're there.
Okay.
They're just, and if they would have not been premature, everything would have been fine.
Okay.
So that's a good point, yeah. They don't lack type 2 cells, no. They're just not ready to work yet because they don't really get ready to work until near the end. Okay, so let me do this and show you the logic. Okay, this fluid directly surrounds, is in contact with cells in tissue. Wow, look what appropriate. The first one happens to be right? Interstitial, but you should always look at all of them to see why they're wrong. Okay, well, first of all, blood doesn't surround cells, right? It's interstitial fluid that surrounds cells. Lymph. The fluid in between cells is called interstitial fluid. It's not called lymph until it gets into the lymph vessel. Interstitial fluid will become lymph, right? Renal, I don't know why I put that in there, it means kidney. Hepatic means, and now look at this. If you didn't know renal, doesn't mean, if you didn't know that, you better put that in your notes because I said it in class, it could be on tomorrow's assessment, right? Anything I do in class, except the dog names we talked about yesterday, I want to ask you about that. Anything. Renal is kidney, hepatic is liver, perilymph. That's a word I made up, but I got to tell you about peri. Peri means around. P-E-R-I. I'm not sure if there's even a word called perilymph. I made it up. Sometimes it gets late. I gotta have a word. I gotta show you on one look how I can make up words. Okay, I gotta show you that. So then now in your notes, you could write, so from this, knowing this is the right answer, you could write in your notes, interstitial fluid directly